Okay, so this will be our week 4 lecture, and our objectives for this week will include describing the relationship among metabolism, catabolism, and anabolism. Also, you should be able to look at and understand the different components of an enzyme, what an enzyme is made up of. In addition to that, you should be able to explain the relationship between a genotype and phenotype. Also, you should be able to define amphibolism, and you should also be able to differentiate between the different types of genetic mutations or gene mutations. Metabolic pathways are the series of chemical reactions that occur within a cell, and they're often determined by coenzymes. Enzymes are specialized proteins that speed up, or the term that we use is catalyze, specific chemical reactions in a cell. Enzymes, they are encoded by genes, which are parts of the DNA, segments of the DNA that contain the instructions or the recipe for making a specific protein. So therefore, the presence or the absence of a particular enzyme can be determined by the genes that are present in that organism's genome. Amphibolism. This refers to the ability of a metabolic pathway to integrate both catabolic as well as anabolic pathways, which will allow for the efficient use of resources within a cell. Catabolic pathways break down larger molecules into smaller ones. Okay? And when this happens, it releases energy in that process. On the other hand, anabolic pathways use that energy to build up larger molecules from smaller ones. Okay, so to give an example, if you have a disaccharide or a polysaccharide for that matter, okay, a large sugars, and you break them down into glucose monomers, that would be going from large to small. Okay, so this is an example of a catabolic pathway. Right? We're taking large molecule and making it small. On the other hand, when you take amino acids and you assemble them together to make proteins, that would be an example of anabolic pathways. So by integrating both types of pathways, the cell is able to balance its energy needs and avoid wasting resources. So again, enzymes are biological catalysts that are able to speed up chemical reactions in living organisms. They're highly specific for a particular chemical reaction, and they're not used up in the process. Okay, so instead they end up being recycled, they get reused. So enzyme is typically made up of a protein component called an apoenzyme. Okay, and it also is made up of a non-protein called a cofactor. Okay, so again, enzymes made up of two parts, an apoenzyme as well as a non-protein part called cofactor. Right? So a cofactor could be either organic or it could be inorganic, and if it's an organic molecule, we call that a coenzyme. Okay? So when the proper apoenzyme and a cofactor combine together, they form a functional enzyme known as a haloenzyme. Okay? So the cofactor may be tightly bound to the apoenzyme, or it may only transiently associate with it during that reaction. So the presence of cofactors, especially coenzymes, can be critical for enzymes to be able to function properly. Okay? Why? Because again, they can serve as electron carriers, or they can even provide important functional groups that help facilitate that chemical reaction. So over here, again, we have an apoenzyme. Now we take this apoenzyme, remember this is the protein part, it's inactive. When we combine that with a coenzyme, now we have a haloenzyme or a whole enzyme. This is an active enzyme. Okay, and what they're showing is, okay, fine, you take this apoenzyme and then you combine it with this coenzyme, they fit together. Then you have a substrate, something else that this enzyme will interact with to speed up the reaction. Okay? All right, let's take a closer look at some of these important coenzymes. So first one up is NAD+, which is nicotinamide adenine dinucleotide. We also have its phosphorylated form, NADP+. These are involved in redox reactions where they serve as electron carriers. NAD is typically involved in catabolic pathways, while NADP+ is involved in anabolic pathways. The next one, FAD, or flavin adenine dinucleotide, is another redox coenzyme that plays a role in energy production by participating in the electron transport chain. Okay? It's also involved in various oxidation and reduction reactions as well. Then we have the last one, coenzyme A. Coenzyme A is involved in various metabolic pathways, including the breakdown of fatty acids, the citric acid cycle, also the synthesis of fatty acids and cholesterol. Coenzyme A plays a critical role in the transfer of acetyl groups, which are important in energy metabolism. Okay, so these coenzymes that we've discussed, they're critical for enzyme function, as well as they're often used in the transfer of electrons or functional groups during metabolic reactions. And what they do is, very simplistically to explain, they're going to allow for efficient transfer of energy and substrates between different metabolic pathways. And this allows the cell to carry out a wide range of metabolic functions. Enzymes can be classified based on the type of chemical reactions they catalyze. And the following are the 6 main classes of enzymes. Okay? So the first one you have over here are oxidoreductases. Okay? If you look over here, oxidoreductase. Okay? So these class of enzyme, they catalyze oxidation reduction reactions. Okay? And this is where electrons are transferred from one molecule to another. And examples of oxidoreductase include dehydrogenases as well as oxidases. Transferases, okay, the next one. These enzymes, they catalyze the transfer of a functional group such as a methyl or a phosphate group from one molecule to another. Example of this would include kinases and transaminases. Okay, next we have the hydrolases. These enzymes, they catalyze the hydrolysis of a bond, okay? Typically using water to break the bond. So examples of hydrolases include lipase and proteases. Now, Lyases, these enzymes catalyze the removal of atoms or groups of atoms from a molecule without using water to break bonds. So examples of lyases include decarboxylases as well as dehydratases. Okay. Next we have the isomerases. These enzymes catalyze the rearrangement of atoms within a molecule to form an isomer. Examples of isomerases include epimerases and mutases. Finally, we have the ligases. Ligases are enzymes that catalyze the joining of two molecules, typically by forming a new bond and using ATP as a source of energy. So examples of ligases include DNA ligase and ATP synthase. Now, let's look at the 4 main factors that can influence enzyme activity. First, we have temperature. Right? Enzyme activity is generally optimized at a particular temperature, known as the enzyme's optimal temperature. And this is because enzymes are sensitive to temperature changes, and high temperatures can cause them to denature, or that means to lose their 3D shape, and then they become inactive. Also, we have pH. So enzymes also have an optimum pH range, and this is the pH at which they are most active. So changes in pH can alter changes on the amino acid residues within the enzyme, and this also will affect its shape, hence its function. Okay. Next, we have the substrate concentration. So the rate of an enzyme catalyzed reaction can increase with increasing substrate concentration up to a certain point. Now, this point is what we call the saturation point, and this is where the enzyme is working at its maximum rate and all active sites are occupied. Okay. Then we have inhibitors. Now, enzyme activity can be inhibited by molecules that bind to the enzymes, blocking its active sites and preventing substrate binding. Now, inhibitors can be reversible or they can be irreversible, and they can be competitive or they can be non-competitive with respect to the substrate. So if you look over here, and they're showing you that, look, at high temperatures or high or low temperatures, at specific temperatures that are not optimal for a protein, it will start to denature, or again, it loses its shape. So remember what I said, structure and function, they always go together. Okay, so if you lose the structure, now this is no longer functional to do whatever its job is. All right? When you're looking over here, this is showing, again, this would be the optimal temperature of this enzyme, whatever this is. And if you look down over here or over there, you can see that, again, at these temperatures, this is the activity level of the enzyme. It's not at its optimal. So it's doing a lot of work over here at this temperature range, which is between 35 to 40 degrees. It's doing a lot of work. Okay, but then you go below that, it's doing much less. Okay, below this temperature range or higher, greater than that, it doesn't function as well. Okay, it's not doing as good of a job as it can. And the same will go for pH. So when you look at pH, again, what are we seeing? That at, first of all, again, you have between 4 and 6, you would say, or maybe between 4 and 5 would be the optimal pH for this enzyme. And then you go below or above this pH, the activity starts to decrease, okay, as you can see by the graph. Also, again, it's telling you substrate concentration. So when you start increasing the concentration of the substrate, right, the rate of the reactions, it will start to increase. So when you have lower amounts of substrate, enzyme's not that active. When you increase it, now it's active. Okay. So again, at higher substrate levels, you end up having higher activity. Of course, up until a certain point. When you go too high, then the efficiency will drop. Okay. Moving on to bacterial metabolism. So we have two types, autotrophs and heterotrophs. Autotrophs are organisms that can synthesize their own organic molecules, such as sugars and amino acids from inorganic sources. And we have two main types of autotrophs. We have photosynthetic autotrophs and we have chemoautotrophic autotrophs. So the first one, the photosynthetic autotrophs, are those such as plants and some bacteria which use sunlight as their energy source to convert carbon dioxide and water into organic molecules. Now, chemoautotrophic autotrophs, these are certain bacteria, okay, that use energy from chemical reactions such as oxidation of inorganic compounds like sulfurs or ammonia to synthesize organic molecules. Now let's move on to the next type, the heterotrophs. These are organisms that cannot synthesize their own organic molecules and must obtain them from other sources. And we have two main types of heteroautotrophs. There's parasites and saprophytes. Parasites will obtain their nutrients from a living host, and often they cause harm to that host. Okay? Saprophyte, on the other hand, can obtain their nutrients from a dead or decaying organic matter, such as fungi, as well as some bacteria. So again, this table over here kind of goes over what we just spoke about. Again, it tells you their nutritional type, and then their energy source, as well as their carbon source, and some examples. Okay, sugars are a key source of energy for living organisms that can be broken down through different metabolic pathways, including the aerobic respiration and anaerobic respiration, as well as fermentation. So, in aerobic respiration, this is a metabolic pathway that involves the breakdown of glucose or other organic molecules in the presence of oxygen. So you need oxygen. Okay? So oxygen is needed to release energy. And it's a highly efficient process that's able to produce a large amount of ATP. Okay? ATP, remember, it's adenosine triphosphate. And so again, this is what that cell uses for energy. Anaerobic reactions, on the other hand, an anaerobic respiration, so this pathway, the metabolic pathway, involves the breakdown of organic molecules in the absence of oxygen to release energy. It's less efficient than the aerobic respiration that we mentioned, but still it's able to produce some ATP. Okay? Next, we move on to fermentation. This is a metabolic pathway that involves the breakdown of glucose or other organic molecules in the absence of oxygen to release energy. Unlike aerobic and anaerobic respiration, fermentation does not require an electron transport chain, and it also produces a limited amount of ATP. Fermentation is used by some organisms such as yeast and bacteria to produce alcohol, lactic acid, or some other product. The most efficient way to extract energy from glucose is through a process called cellular respiration, which involves the following three main stages. So in the first one, in glycolysis, so again, this is the initial stage of cellular respiration, which takes place in the cytoplasm of the cell. It involves the breakdown of glucose into two molecules of pyruvate, along with the production of a small amount of ATP and NADH, nicotinamide adenine dinucleotide. Okay. Next, we have Krebs cycle. Krebs cycle is also known by a couple of other names. You can call it, sometimes some people call it, they call it the TCA cycle, or you can even call it the citric acid cycle, or again, you can call it the Krebs cycle. It's the same thing, just different names. This is a series of chemical reactions that takes place in the mitochondria of cells, and it involves the conversion of pyruvate to acetyl coenzyme A, and the further breakdown of this molecule to carbon dioxide, while generating more ATP as well as NADH. Then we have electron transport chain. Okay. This is the final stage of cellular respiration, which takes place in the inner membrane of the mitochondria of cells. It involves the transfer of electrons from NADH as well as FADH2, and that's flavin adenine dinucleotide. Okay. By the way, FADH also gets produced over here in the Krebs cycle. I should have said that earlier. So both NADH and FADH ends up being produced in the Krebs cycle. So again, electron transport, it involves the transfer of electrons from both NADH and FADH to a series of protein complexes, ultimately leading to the production of a large amount of ATP. Okay, so overall, cellular respiration is a highly efficient process that allows the cells to extract the maximum amount of energy from glucose. So it'll produce a total of about 38 molecules of ATP from one molecule of glucose. The final electron acceptors in cellular respiration is an important component of the electron transport chain, which allows for, again, as we said before, the transfer of electrons from NADH and FADH to the final electron acceptors, and thus producing ATP. Now, the choice of final electron acceptors can vary depending on the organism and the environmental conditions. So let's look at some of the examples. So it could be a sulfate reducer. And these are bacteria that use sulfate, okay, that's what this is, sulfate, as their final electron acceptor in anaerobic respiration. Then there's the methane reducers. These are archaea that use carbon dioxide as their final electron acceptor in anaerobic respiration, producing methane in the process. There's also the nitrate reducers. These are bacteria that use nitrate as their final electron acceptor in anaerobic respiration. Now, it's true that oxygen is the most oxidizing final electron acceptor, and as a result, aerobic respiration is generally more efficient than anaerobic respiration. And the reason for that is because oxygen and water coupling allow for the transfer of more electrons and the production of more ATP. So, however, anaerobic respiration can still be an important metabolic strategy for organisms living in environments where oxygen is scarce or absent. All right, so as we said before, fermentation is an alternative metabolic pathway for the breakdown of glucose that takes place in the absence of oxygen. And unlike aerobic respiration, which uses oxygen as a final electron acceptor in the electron transport chain, fermentation instead will use organic compounds such as pyruvate or acetaldehyde as the final electron acceptor. So looking over here again, you can see that fermentation can be said to be incomplete oxidation of glucose, meaning that, again, it's only extracting a portion of the energy that could be obtained through the complete oxidation in aerobic respiration. So because it doesn't use the Krebs cycle or the electron transport chain, the net ATP that's yield from fermentation is much lower than what we have seen in aerobic respiration. Now, with that said, fermentation can still be useful for certain organisms that are living in environments where oxygen is scarce or absent, as it allows for the continued production of ATP in the absence of aerobic respiration. All right, so another look at glycolysis. So glycolysis is the metabolic pathway that converts glucose into 2 molecules of pyruvate along with the production of 2 ATP and 2 NADH molecules. Fermentation pathways, again, these are alternative metabolic pathways that take place when oxygen is not available for cellular respiration. Now, there's 2 types of fermentation pathways, but 2 of the most common are the homolactic acid and the alcoholic fermentation. Okay? So there's many different types of fermentation pathways, but these 2 are the most common. So let's take a look at this one first. Okay? The homolactic acid fermentation. So in homolactic acid fermentation, this takes place, we find it taking place in some bacteria, including streptococci and lactobacilli. And also we see this in some human cells. In this pathway, pyruvate is converted into lactic acid, and NADH produced by glycolysis is oxidized back to NAD+, which can be used in glycolysis to produce more ATP. For alcoholic fermentation, this takes place in yeast and some other microorganisms. In this pathway, pyruvate is first converted into acetaldehyde, which is then reduced to ethanol which is again ethyl alcohol, by NADH. Like homolactic acid fermentation, this pathway allows for the regeneration of NAD plus to keep glycolysis going. Both of these pathways, okay, homolactic acid fermentation and alcoholic fermentation, they're less efficient than aerobic respiration in terms of the ATP production. But again, as we mentioned before, certain organisms, they can use it to survive, to continue to produce ATP when oxygen levels are scarce or absent. Moving on to mixed acid fermentation. In this pathway, we find it in some bacteria, including E. coli and some other enteric bacteria. Again, enteric bacteria are gut-living bacteria that you find living in the gut. In this pathway, pyruvate can be converted into several different organic compounds, including lactic acid, acetic acid, hydrogen gas, carbon dioxide, succinic acid, and even ethyl alcohol. The pathway is called mixed acid because it produces a mixture of different organic acids. And this pathway is important for some bacteria that live in the human gut, as well as other environments. Next, we have butylene glycol fermentation. Okay, butylene glycol fermentation. This pathway occurs in some bacteria, including Pseudomonas species. In this pathway, pyruvate ends up being converted into a compound that's called 2,3-butyladenol. Okay, 2,3-butyladenol can be used by the bacterium for energy or for other metabolic processes. Next, we have propionic acid fermentation. Propionic acid fermentation. So in propionic acid fermentation, this pathway will occur in some bacteria that include propionic bacterium species. Okay, propionic bacterium species. Okay, propionic. If you break down the words, it gets easier to combine and remember also. So what happens over here? In this pathway, pyruvate ends up being converted into propionic acid. Okay? And this is an important chemical for some industrial and agriculture processes. Microorganisms can use a wide range of products through fermentation, including various types of organic acids such as lactic acid and acetic acid, as well as solvents like acetone and butanol. In addition, some microorganisms are capable of producing vitamins, antibiotics, and even hormones through fermentation. So these fermentation processes are often used in large-scale industrial applications to produce these compounds in a cost-effective manner. Non-carbohydrate compounds can also serve as sources of energy for microorganisms. Lipids, for example, can be broken down by lipases into glycerol and fatty acids. Glycerol can be converted into dihydroxyacetone phosphate, which can enter the glycolytic pathway. Fatty acids undergo beta oxidation, which produces acetyl coenzyme A that can enter the Krebs cycle. Proteins can also be broken down by proteases into individual amino acids. The amino groups that are removed through deamination and the remaining carbon compounds will end up being converted into Krebs cycle intermediaries. Okay, so these pathways, they allow microorganisms to catabolize a wide variety of compounds and generate energy for their metabolic needs. The genome of an organism includes all of its genetic material, including all of its genes and non-coding regions of DNA. The genome, we find it contained within the chromosomes of the cell. Again, chromosomes are structures that are made up of DNA and proteins. Different organisms have different numbers and sizes of chromosomes, and the DNA sequences within the chromosomes also will vary among the different species. So the study of genomes and the genetic information they contain is known as genomics. And it has important implications in the fields such as medicine, in the fields such as agriculture, as well as biotechnology. Some genes encode functional RNA molecules such as transfer RNA, which we can abbreviate as tRNA, or rRNA, which you can abbreviate, again, which stands for, not abbreviate, which stands for ribosomal RNA. These are involved in protein synthesis. Additionally, some genes have regulatory functions that control the expression of other genes, or they play a role in processes such as DNA replication or DNA repair. So, what is a gene? So, a gene is a segment of DNA that contains the instructions for the development, function, and reproduction of living organisms. These instructions are encoded in the sequence of nucleotides in the DNA molecules and are passed from one generation to the next through the process of reproduction. Now, the information that's contained in genes determines an organism's traits. This includes its physical characteristics, behavior, and even susceptibility to disease. Phenotypic properties are the observable characteristics of an organism, while genotype refers to the genetic makeup of an organism. Okay? Genotype, what are the actual genes? Phenotype, what does it produce? What does it look like? Okay? So again, observable characteristics, phenotype. Okay? Genotype is the genetic makeup. What are the instructions? What's the code? The code is the genotype. So variations in phenotype are caused by changes in the genotype. Okay? And this can result from mutations, recombination, or other genetic mechanisms. And these changes can affect the sequence of amino acids in a protein, the regulatory elements that control gene expression, or the overall organization of genes on a chromosome. All right, so we have terminology here that you should all be familiar with. Again, what is genetics? What are genes? What are chromosomes? What's the genome? Be sure you know what all these are. Okay, you're expected to know this. Again, additional terms, and we've mentioned already, phenotypes, genotype, and genomics. So again, genotype of an organism determines its phenotype, but not all genes are active at all times. The expressions of genes can be influenced by various factors such as environmental conditions, developmental stage, as well as regulatory mechanisms within a cell. So gene expression can also be altered by mutations or changes in the DNA sequence. And this can result in changes in the phenotype of an organism. All right, looking at the prokaryotic chromosome. Okay. Prokaryotic chromosomes are the main portion of DNA in prokaryotic cells, along with the associated proteins as well as RNA. Prokaryotic cells are haploid. This means that they have a single copy of the chromosome, and the typical chromosome is a circular molecule of DNA that's located in the nucleoid region of the cell. Now, in addition to the chromosome, prokaryotes may also have plasmids. Plasmids are small, circular, double-stranded DNA molecules that can replicate independently of the chromosome. Okay? Plasmids may contain genes that confer some advantageous traits to the bacterium, such as antibiotic resistance or the ability to metabolize certain nutrients as well. So again, you can see over here, this is the nucleoid, okay, within this prokaryotic cell. And again, over here, you can see that you got a bacterium over here, And here's some genetic material that is identified. And over here we can also see some plasmids over here as well. Alright, so as I said, plasmids are small, circular, and they can even be linear, double-stranded DNA molecules that exist independently of the chromosomal DNA in prokaryotic cells. Again, they're not essential for normal growth, metabolism, or reproduction of the host cell, but they can confer certain advantages to that host cell under certain conditions. Plasmids, they can replicate independently, and they could be transferred between cells through various mechanisms, including conjugation, transduction, as well as transformation. And there's many types of plasmids, and each, they have their own set of genes and functions. Some plasmids are fertility factors, which carry genes for the transfer of plasmids from one bacterium to another. And this happens through conjugation. Resistance factors, or R plasmids, they carry genes that give resistance to antibiotics or other antimicrobial agents. Bacteriocin factors can carry genes for the production of bacteriocins, which are proteinaceous toxins that can kill or inhibit the growth of closely related bacteria. Virulence plasmids will carry genes that are involved in the virulence, or another term would be the pathogenicity of the host cell, allowing it to cause disease in a host organism. Alright, eukaryotic cells can be diploid or polyploid depending on the organism and the stage of the life cycle. For example, human cells are diploid. Again, this means 2 copies of each chromosome, while some plant cells can be polyploid, meaning, again, they have multiple copies of each chromosome. Some eukaryotic microorganisms, such as fungi, algae, and protozoa, can carry plasmids, just like prokaryotes. These plasmids can carry genes that give selective advantages, such as, again, antibiotic resistance or the ability to produce certain metabolites. Additionally, some plants carry plasmids in their chloroplast. Now, however, it's important to note that plasmids are much less common in eukaryotes than they are in prokaryotes. Okay, DNA is a polymer of nucleotides, with each nucleotide consisting of a sugar, a phosphate group, and a nitrogenous base. Now, the double helix structure of DNA is formed by 2 complementary strands of nucleotides that are held together by hydrogen bonds between the nitrogenous bases, adenine pair with thymine, cytosine pairs with guanine. Okay, so remember, adenine always will pair with thymine. A always goes with T, as you can see here. And G always goes with C, as you can see here. Okay? And the backbone of each strand is made up of alternating sugar and phosphate groups. So you can see sugar, phosphate, sugar, phosphate, sugar, phosphate. Same here. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Notice one is going in a 3' to a 5' direction, while the other goes in a 3 to a 5' direction. So, again, these strands, they run in opposite direction. Again, and we call this term of going from one direction to the other direction is said to be anti-parallel. Okay, so the strands run in okay, so because the strands are running in opposite directions, we say that they are anti-parallel. Okay, and this is associated with proteins that are called histones to form chromatin. Okay. So during DNA replication, two strands of the DNA double helix separate, and each strand serves as a template for the synthesis of a new complementary strand. This process is semi-conservative, okay, because each new DNA molecule is composed of one original, or again, one parent strand, and one new strand, one newly synthesized daughter strand. It can be said, okay, a daughter strand. This is the new one that gets produced. So the synthesis of new DNA strands is an anabolic polymerization process, and this requires monomers in the form of nucleotides and energy in the form of ATP, adenosine triphosphate. Okay, so over here you can see the original DNA, and what ends up happening is this original DNA, it will open up, okay, it splits, and then when that happens, then you start to make new DNA. So if you look over here, the purple is the original strand, okay, and then you see the new strand, or the daughter strand is over here, okay, and that is the this aqueous color, this light blue or green, whatever you see this as, and then the purple, the darker one, that's the parent strand, or again, the original strand, you can call it. So essentially you take this and now you end up having what? From one DNA, now you got two molecules. And when these divide as well, you end up having, look, 4 DNA molecules now. Okay? Alright, so replication starts at points that are called origin. Okay? So the leading strand is synthesized continuously in the 5' to 3' direction, while the lagging strand is synthesized discontinually in short fragments that are called the, let's see, it's not written over here. All right, it's called Okazaki fragments. Okay, Okazaki, Okazaki fragments. Okay, and that also goes in the 5' to 3' direction. Okay, the Okazaki fragments are then joined together by DNA ligase, okay, to form a continuous strand. Right, so once again, let's go through this. We said that replication will start at points on DNA that are called origin. We said that the leading strand gets synthesized in a 5' to 3' direction. And we said the lagging strand isn't synthesized, isn't made continuously. Instead, it's made in fragments. And these fragments we said are called the Okazaki fragments, which also gets produced in a 5' to 3' direction. Okay? So, and then we said that these Okazaki fragments, they end up being connected by DNA ligase to form a continuous strand. Eukaryotic DNA replication is similar to bacterial replication, but it has some differences. For example, eukaryotes use 4 DNA polymerases instead of 1 that is present in bacteria. And they have thousands of replication origins instead of a single origin. Okay? So in bacteria, there's 1 replication origin. In eukaryotes, we have multiple origins, lots of origins. Additionally, eukaryotic Okazaki fragments are much shorter than those in bacteria. Why? Again, we have so many more origin points, therefore there will be shorter Okazaki fragments. In addition to that, in eukaryotic cells, cytosine bases are methylated, while in bacterial cells, adenine bases are methylated. So again, over here, this table, it's going over some of the differences that we'll observe in genomes amongst bacteria, archaea, and eukarya. All right, the central dogma of genetics states that genetic information can flow from DNA to RNA to protein. Okay, so DNA ends up leading to the production of RNA, and RNA ends up to the production of proteins. DNA is transcribed into RNA in a process called transcription. Transcription takes place in the nucleus of eukaryotic cells, or in prokaryotic cells, it will take place in the cytoplasm. RNA transcript then leaves the nucleus and is translated into a polypeptide chain on ribosomes in the cytoplasm. And this is called translation. So the sequence of amino acids in the polypeptide chain is determined by the sequence of nucleotides in the RNA molecule. So transcription is the process by which genetic information encoded in DNA is copied into RNA molecules, such as messenger RNA, transfer RNA, as well as ribosomal RNA. So during transcription, RNA polymerase binds to a promoter sequence on the DNA strand, and then it reads the DNA template strands in the 3 to 5 prime direction. And it will synthesize RNA in the 5 to 3 prime direction. The transcription will continue until RNA polymerase reaches the terminator sequence. And this gives a signal to stop, meaning it's the end of transcription. So if you look over here, all right, we have a strand over here, okay? And so when RNA is being produced, this is the strand that's going to be read, okay? So again, from T we get A. Now in RNA does not have T. There's no thymine. Instead, there's uracil present, okay? So again, remember, we have complementary base pairs. Remember what we said, A always goes with T, right? A always goes with T, or again, A always goes with T, doesn't make a difference. T goes with A, they always go together. G is always with C, or C with G. So when you look over here, remember we're reading this. T will produce A, okay? A, remember what we said, we're supposed to have T, but remember in RNA, there's no T presence. What do we have instead? U, uracil. So here's U. And then C will give us G. G will give us C. C will give us G, and on and on and on. So this essentially will say, okay, you start over here, and then it ends up saying this 1, 2, 3. These three act as a 1, 2, 3 over here. These three bases will act as a code, right? So you read this code, and this code translates into the amino acid methionine. Then you have another one. This translates into arginine, and this translates into tyrosine, and this will translate into leucine. So this goes on and on and on until on this RNA sequence or on the DNA, it comes to a stop region. So then again, that's a terminator, and it stops copying at that point. So again, remember, this is the genetic code, all right? This genetic code ends up, or again, this is the genome. So this produces this sequence of amino acids, which again is a polypeptide or a protein. And then this ends up bringing upon some type of a trait, right? It looks like something, and that we call the phenotype. All right, RNA primers. RNA primers are short sequences of RNA that serve as starting points for DNA synthesis. In DNA replication, RNA primers are synthesized by primase and are necessary for DNA polymerase to start replication. In DNA repair, RNA primers are used to initiate the removal of damaged DNA. Messenger RNA, okay, or mRNA, carries the genetic information from DNA to ribosomes, where it serves as a template for protein synthesis. The sequence of nucleotides in mRNA is determined by the sequence of nucleotides in the DNA template. This is what we saw in the previous slide. Now, rRNA, ribosomal RNA, is a structural component of ribosomes, which are the cellular machinery that's responsible for protein synthesis. So this is where protein assembly takes place. Okay. So along with ribosomal proteins, rRNA forms two subunits of the ribosome. Okay. Moving on to tRNA. tRNA or transfer RNA is responsible for carrying amino acids to the ribosome during protein synthesis. Each tRNA molecule binds to a specific amino acid and recognizes the corresponding codon on the mRNA. Okay? So it's bringing the correct amino acid to the ribosome assembly point, right, to the RNA. Then we have the regulatory RNA. All right. Regulatory RNA includes a diverse group of RNA molecules that regulate gene expression at the transcriptional or the post-transcriptional level. So examples include microRNAs, small interfering RNAs, as well as riboswitches. These molecules are able to bind to mRNA and prevent it from being translated, or they'll even degrade mRNA. And so again, they have the capacity to regulate the amount of proteins that are produced. All right, so transcription occurs in the nucleus of eukaryotes, and there are 3 types of RNA polymerases that transcribe different types of RNA. Transcription factors are also involved in regulating gene expression. Messenger RNA or mRNA will undergo several processing steps, including capping, splicing, as well as polyadenylation, before it can be transported out of the nucleus and translated into protein. All right, so during translation, the genetic information in mRNA is used by ribosomes to direct the synthesis of a specific polypeptide chain. So this process involves the coordinated interaction of tRNA molecules, which carry amino acids, to the ribosome and mRNA to make sure that the amino acids are added in the correct sequence to form the desired polypeptide chain. So AUG, okay, AUG is the start codon, which codes for methionine and signals for the beginning of translation. Whereas UAA, UAG, as well as UGA are the 3 stop codons which signal the end of translation and do not code for any amino acids. Okay, so you want to start translation, we look for the start codon. Okay, and again, what did we say? AUG is the start codon, okay, and it codes for methionine. For stop, we have 3 of them over here, right? UAA, UAG, as well as UGA. Now, the genetic code is said to be degenerate. What does that mean? It means that more than one codon can encode the same amino acid. So for example, the amino acid leucine can be encoded by 6 different codons. There's UUA, UUG, CUU, CUC, CUA, as well as CUG. Now each tRNA molecule carries a specific amino acid on one end and a complementary anticodon on the other end. The anticodon base pairs with the corresponding codon on the mRNA during translation, which allows the correct amino acid to be added to the growing polypeptide chain. Okay. So messenger RNA, transfer RNA, and ribosomal RNA, okay, mRNA, tRNA, and rRNA. Messenger RNA carries the genetic information from the DNA to the ribosome, where it's read and translated into a protein sequence with the help of transfer RNA, which carries amino acids that are added to the growing protein chain. So the ribosomes that are made up of ribosomal RNA and protein catalyze the formation of peptide bonds between the amino acids to create the protein. All right, looking at translation, the events in translation can be broken down into 3 steps, or 3 stages, we can say: initiation, elongation, as well as termination. By the way, these 2 steps, initiation and elongation, they require energy in the form of guanosine triphosphate, or GTP. So let's go on to the first stage. So during initiation, we have a ribosome that will form, okay, to the messenger RNA. Now, you have to keep in mind that the ribosome is made up of 2 parts, a small subunit and a large subunit. So what's the first thing that will happen? All right, so the first step during initiation, the small ribosomal subunit will attach to the mRNA molecule. And then the initiator tRNA that's carrying methionine binds to the start codon. Again, this is that AUG. Then the large ribosomal subunit will come and join the small one, and it forms the complete ribosome. Okay. Now, the next part, during elongation, the ribosome moves along this mRNA molecule, adding 1 amino acid at a time to the growing polypeptide chain. All right. So, as the polypeptide chain is growing, how is this happening? It's happening because each amino acid is brought to, or is carried to, the ribosome by a specific tRNA molecule, okay, which base pairs with the complementary codon on the messenger RNA. Now we move on to termination. So during termination, the ribosome reaches a stop codon. And remember what we said, stop codon could be UAA, UAG, or UGA. Okay, so during this stage, right, during termination, ribosome reaches the stop codon on the messenger RNA. And when that happens, the polypeptide chain ends up being released from the ribosome. The ribosome then breaks down into its subunits, and the mRNA ends up being released. Okay, quorum sensing is a process that's used by bacteria to communicate and coordinate group behavior based on the density of their population. Bacteria produce and secrete small signaling molecules called autoinducers that accumulate in the environment as the population grows. Now, once the concentration of autoinducers reaches a threshold level, the bacteria can detect it and initiate a response, such as the expression of certain genes. This mechanism is often used to regulate the expression of genes involved in virulence, biofilm formation, and even antibiotic resistance. So, for example, the bacterium Pseudomonas aeruginosa uses quorum sensing to regulate the production of its virulence factor, such as enterotoxin A and elastase. Now, in the absence of a high cell density, these genes, they're not expressed, and this allows the bacteria to conserve energy. However, when the population density reaches a certain level, the bacteria starts expressing these genes that will, again, allow, and it leads to the production of virulence factors that allow the bacteria to attack the host. All right, mutagens can be physical or chemical agents that cause mutations in DNA. Physical mutagens include ionizing and non-ionizing radiation, while chemical mutagens include nucleotide analogs and nucleotide-altering chemicals. Nucleotide analogs can disrupt DNA and RNA replication by incorporating themselves into the growing nucleic acid chain. Nucleotide-altering chemicals are able to alter the structure of nucleotides that result in base pair substitutions. All right, frame shift mutations can cause a change in the reading frame of the mRNA, and this leads to a completely different amino acid sequence downstream of the mutation. So this can result in the production of a non-functional or a partially functional protein. So as a result, frame shift mutations are often deleterious and can have serious consequences, including cancers. So some examples of frame shift mutagens include benzopyrene in smoke, as well as aflatoxins. Ionizing radiation has high energy and can penetrate deep into biological tissues. When it interacts with atoms in DNA molecules, it can ionize them, causing the formation of free radicals and reactive oxygen species, or ROS. And these are able to break the phosphodiester bonds between the nucleotides. And this ends up resulting in a physically broke down DNA. Okay? So the damage that's caused by ionizing radiation can lead to mutations, chromosomal abnormalities, and even cell death. All right. In terms of the frequency of mutations, mutations are rare events, and the frequency of mutations will vary depending on the organism and the type of mutation. Generally, the mutation rate is about 1 in every 10 million genes, okay, which is still significant considering the number of genes in an organism. Mutagens can increase the mutation rate by a factor of 10 to 1,000 times, which can have serious consequences for the organism. However, many mutations, they don't have a significant effect on the organism, and some can even be neutral or beneficial for that matter. Now, nonetheless, mutations that result in non-functional proteins or stop transcription can have a negative impact on the organism's survival as well as their reproduction. Recombination is the process by which new combinations of nucleotide sequences are generated in DNA molecules. This often occurs between homologous sequences, such as during crossing over events in meiosis. Recombination can also occur between non-homologous sequences, which can result in the creation of new genes or the transfer of genetic material between different species or even kingdoms. So, recombination plays an important role in evolution by generating genetic diversity, which can be acted upon by natural selection. Right, vertical gene transfers. Vertical gene transfers is the transmission of genetic material from one parent to offsprings during reproduction. The process occurs during the replication and division of cells, and this is what's responsible for passing on genetic information from one generation to the next. And it's the primary mechanism for maintaining genetic continuity within a species over time. Okay, horizontal gene transfer is an important mechanism of genetic variation and evolution in prokaryotes. We have 3 types of horizontal transfer. We have transformation, transduction, as well as bacterial conjugation. Let's look at the first one. Transformation, we have the uptake of free DNA from the environment by a competent bacterial cell. The DNA is able to integrate into the recipient cell's genome through recombination, and it can bring about new traits or functions. For the next one, in transduction, we have the transfer of DNA between bacteria by a bacteriophage. Okay? A bacteriophage or a phage is a virus that infects bacteria. Okay? So during infection, the phage may accidentally package host DNA instead of its own and transfer it to another bacterial cell upon infection, which will lead to genetic recombination. Okay? Finally, we move on into bacterial conjugation. This is a mechanism of DNA transfer in which the genetic material is transferred from one bacterium to another via a pilus, which is a protein appendage. If you remember back, a couple of lectures ago, we talked about pili. So again, revisit that if you do not recall. So moving along with this, with conjugation, the donor bacterium, it possesses a conjugative plasmid that contains the genes for the pilus and the DNA transfer machinery. The plasmid replicates and is transferred through the pilus to the recipient bacterium. So conjugation can transfer genes for antibiotic resistance, virulence factors, as well as some other traits. All right, so for transformation, again, when a bacterial cell is in a state of competence, okay, the recipient cell, it takes up DNA from the environment. So again, competence, this term, is a physiological state of bacteria that allows the cell to take up extracellular DNA. Okay? It's often induced by changes in growth conditions such as nutrient depletion or by exposure to specific chemicals or physical agents. Once the DNA is taken up, it can recombine with the recipient cell's genome, resulting in new genetic combinations. So transformation provides the first evidence, okay, that DNA is the genetic material, right? So transformation ended up giving us the first proof that deoxyribonucleic acid is in fact genetic material. All right, so during transduction, a bacteriophage infects a donor bacterial cell and replicates within it, producing new phages. So sometimes during the process of phage assembly, the fragment of the donor bacterial DNA can become mistakenly packaged within a phage capsid instead of the phage DNA. So when this phage infects a recipient bacterial cell, it can transfer the donor bacterial DNA to the recipient cell. So this transferred DNA can then be integrated into the recipient cell's genome via recombination. So transduction can result in the transfer of specific genes or even gene fragments. And it can act as a mechanism for the spread of antibiotic resistance as well as virulence factors among bacteria. All right. Going back to bacterial conjugation. So bacterial conjugation involves a donor cell that possesses a conjugative plasmid. And the conjugative plasmid contains the genes necessary for conjugation, including those that are encoding for the sex pilus. The sex pilus allows the donor cell to attach to the recipient cell and form a conjugation channel, which is what we see over here. Okay? So through this channel, the donor cell can transfer DNA, such as plasmids or part of the chromosome, to the recipient cell. Okay. So conjugation, it's a common mechanism for the transfer of antibiotic-resistant genes among bacteria. All right. Thank you for viewing, and until our next lecture.